Hello and welcome to GeOMe demo number 2, where I'll talk about querying and downloading data from the database. So what we'll do first is come over to the query page, and let's say I'm interested in the same data that I just uploaded in demo number 1. I want to learn about data that we have from the surgeonfish, Acantharis olivaceus. And you can see it comes back with all of the samples in our database and their locations. And I can zoom in on some of these and find particular sampling localities, which will then show me all of the samples in that locality. I can click on any sample and it will take me to a landing page for that sample that has some relevant metadata, as well as data from the event, from the tissue, and the universally unique identifier. And that's what I can do in the simple search. If I want to do something more advanced, I can switch over to advanced. And you'll see that our database has a fairly simple schema in which event is the parent of sample, which is the parent of tissue, which is the parent of the FASTQ metadata. And I can search on any one of these tables. In this case, I want to search on sample attributes, which would be genus and species. And I'll search again for those same samples. And first, it's going to return to me a table of relevant metadata, including the event, decimal latitude and longitude, phylum, expedition code, and I can also see it in a map. And let's say I'm only interested in samples that come from the coral triangle. In that case, I can draw a bounding box. Just around these samples. And redo my search. And now I've just got those samples from the coral triangle. And now let's say I'm interested in data from cytochrome B. I can search on that. And I get roughly the same samples back. And now, if I'd like, I can download this information as either a CSV archive or as an Excel workbook. The metadata will come down as a workbook. And the FASTA data will come down as FASTA files. With GeOMe, we typically refer to Sanger sequence data as FASTA data, as these are the data that don't come with FASTQ with quality scores. And you can see I can pull down all of the metadata that have been submitted for these samples in an Excel spreadsheet. And I can also look at my FASTA file, where I also have a CSV output for the metadata and FASTA data for each sample. Now, if my interest is in something at the genomic scale, I can turn this off and look only for samples that have data that are in the sequence read archive. So if I search for those, I'll get back just 19 results, which I can still see on the map. Here they are. Go to the landing page and see all of the relevant information. And if I want to redo my search, where I search on the FASTQ entity, I can then do this, and it will come back with the relevant accession numbers that I can then take into SRA tools and download those particular data. Okay, now let's go to the R package. GeOMe comes with an R package called GeomeDB. So I can install that package from GitHub. And there's a few different query where I can see all of the different projects that are in GeOMe. I can query the metadata. Choosing from one of the 4 tables or entities that we discussed earlier. In this case, sample. And then the query itself is a lot like SQL. And I get all the metadata back in a data frame. I can perform the same query for FASTA or Sanger sequence data. Except I will ask for data from the SITB marker. And you can see I get a DNA bin object that can then be used in various R packages. Here's all the data. I can also run my query for FASTQ metadata. In this case, I have to specify a project. In this case, I'll specify the diversity of the Indo-Pacific project, which is project number 1, because FASTQ metadata is a child of sample. And I will add to my query that I only want samples that have data in the SRA system, only have registered biosamples in the SRA. And you can see I get back all of the original material sample ID, as well as the original file names and accession numbers, with the run accession number being the one that I would most likely take to the SRA toolkit to use to download all of this data. Okay, with that ends this demo on querying and downloading data from GeOMe. I hope this has been useful for you, and thank you for your attention.